Now we're going to focus on my favorite tissue, muscles. And this mini lecture is going to focus in particular on how muscles generate force. As you now know, muscles are, along with neurons, excitable tissues. So they deal with electrical transmissions, but their interesting trick is to take those transmissions and to generate tension. So the question for this lecture is, how do muscles transform information into force? How did they do this? This is their special property. How is it that that occurs at a molecular level? The conversion of voltage changes into tension. Let's cover some basics. There are three categories of muscle. The one that we're going to focus on first is skeletal muscle. And that's the muscle that's attached to your skeleton. So when you think of muscle, this is probably what you have in mind. It's generally what you eat. Steak, chicken, fish, those are all skeletal muscles. They are controlled with neurons that you are aware of. Okay, we're going to, in a bit, get into the various ways that your body controls things. And the motor neurons are the particular type of efferent neuron that sends transmissions to skeletal muscles. Okay, so that's a somatic pathway. And when you look at the cells of skeletal muscles under a light microscope, they have a crisscross or cross-striated pattern. And they share that feature with cardiac muscle. So you only find cardiac muscle in the heart, and all of the muscle in the heart is cardiac muscle. And it's somewhat similar to skeletal muscle. So it does have that cross-striated pattern. It's located in the heart. But we are not conscious of our body's control of that cardiac muscle. You don't think, all right, I'm going to beat my heart now and then activate those muscles the same way you do your biceps muscle. Finally, we have smooth muscle. Smooth muscle lines the internal organs. So when your stomach rumbles, that's generated by smooth muscle. And it's only indirectly that you become aware of that. You might feel that or hear that. But again, like the cardiac muscle, smooth muscle is controlled by the autonomic nervous system. We'll tell you lots more about the autonomic nervous system in a bit. But just be aware that it's not something that you're conscious of as your body controls those muscles. And it does not have that cross-striated pattern. So if you take smooth muscle under a light microscope, it has a much more diffuse pattern. It has fibers, but they run in all different kinds of directions. So we're going to talk in more detail on cardiac and smooth muscle later, not so much in this and the next lectures, which are solely focused on skeletal muscle. Okay, so let's start to consider skeletal muscle by taking a detailed look at its structure. So this is my very crude rendering of a biceps muscle, and we're going to dissect it virtually. So we sort of take it out. Sorry for the poor drawing. It looks like a sausage, but we take a cross section through a muscle. And you can do this if you're not vegetarian, or even if you are a vegetarian, you could do a dissection. You just cook a piece of meat and chop it open. And what you'll see is that there are these macroscopic fibers. Think of like breaking up tuna fish in a bowl. And those fibers are called fascicles. Don't worry about that term, but they're macroscopic. And those fibers are in turn composed of microscopic fibers. Okay, those are just called fibers. The term fiber refers to a skeletal muscle cell. Okay, so when I say fiber, just think, okay, that's a cell level of organization. Skeletal muscle fibers can be quite large, and one of their interesting features is they can have multiple nuclei. So you can have, let's just say, a muscle fiber running the length of your quadriceps muscle. That's a really long cell, and it has nuclei all along the way. Now let's continue our dissection at a microscopic level and look at what's inside of a single muscle cell. So we zoom in there, And we've got this. Okay, so this is the surface of the cell. By the way, motor neurons terminate, the axon terminal for a motor neuron terminates on the surface of individual muscle fibers. So we'll talk about that in a little bit, but that is, just imagine there's a big motor neuron that terminates right here to activate the cell. If we dissect further, we see more fibers. This is within a cell or a subcellular level of organization, and those fibers are called myofibrils. So myofibrils have a bunch of proteins, which are also very fiber-like or filament-like, and that's what we see here through the cross-section of a bunch of myofibrils. Now, around the myofibrils, there are these structures called T-tubules, and it's this network of structures that emerge from the surface of the cell. There's a pore here that's continuous with one T-tubule, and you can sort of follow the network of T-tubules into the interior of the cell. They're all continuous little portals with a opening at the surface. And closely associated with the T-tubules are another organelle that wraps around the myofibrils. And those are shown in blue here. They're called the sarcoplasmic reticulum. They're related to the endoplasmic reticulum, but we'll see that they have a very special function for muscles. All right, so let's continue our dissection at even a lower level of organization by extruding one of these T-tubules, like that. And let's just take a look at the association of the sarcoplasmic reticulum around the T-tubule. We can see that it has this wrapping shown in blue there. Again, a very close association, not just with the T-tubules, but also with the myofibril itself. It's really, the sarcoplasmic reticulum is an intermediate structure in between the two. And I'm going to tell you all about the function of these structures in a moment. The myofibrils, if you dissect at that level, so we're looking at suborganellar level of organization, that's where you can interrogate the proteins that are within that. Okay, that is the machinery for generating tension. And there's a very characteristic arrangement of these proteins with that crisscross pattern. This is what makes it cross-striated under a light microscope. And that arrangement of proteins repeats in series. So we see a unit right here, and that unit is known as a sarcomere. And that sarcomere is attached to the end of another sarcomere, and then there's another sarcomere on the end of that. So sarcomeres are arranged in series down the length of a myofibril. Now we can dissect even further and look at those proteins that compose the sarcomere. If we do that, we can see that there's two major kinds of structures. They're thick and they're thin. And the thick we show here in this greenish color, show it in blue later. We'll get into its function later on, but it is indeed thicker than the thin filaments. But the thick and thin filaments you'll see are overlapping here. And if we zoom in on just a pair of thick and thin filaments, then we can see that there are these structures that connect the thick filament to the thin filaments, both up top here and down below there. Okay, so that's the structure, and I want you to be familiar with all of the parts that I labeled. There's plenty of other proteins. There's structural proteins. There's an elastic protein called elastin. There's lots of other structures here that I didn't mention, and we're not going to focus on. But to summarize the structures that really matter, we have, again, at the organ level, that's called a muscle. Okay, so if I refer to a particular muscle, just think, okay, that's an organ. And then we had fascicles, don't worry about that term, but below the muscle level we have cells. Okay, so they're called fibers. Within those cells, there are three organelles that I'm focusing on: the myofibrils, the T-tubules, and the sarcoplasmic reticulum, or the SR. The myofibrils, in turn, are organized around sarcomeres, which are these complexes of proteins, and those proteins include the thick and thin filaments. Okay, so that's what I want you to know about the structure. And as we'll see, the structure is really important to understanding how it functions. All right, so let's step through that. Here we're looking at the interior of one muscle fiber. And I mentioned that there's a motor neuron, you can imagine, just sort of slapped onto a muscle fiber. And that's what we see right here. So here's a motor neuron. And what you can see here is that the membrane for this muscle fiber has a sort of an elaboration that serves to enhance its surface area. That's known as the motor end plate. And what we can see here is that the T-tubule, recall that there's those pores for the T-tubules at the surface of the cell. Those pores are openings into the T-tubule. And so if you follow the cellular membrane, then you can run into the interior of the cell along the T-tubule. So the cell membrane at the surface is continuous with the T-tubule. So our big question here is, how does this cell take an excitatory signal, that's our information, and convert it into tension, the generation of force? And this is a 7-step process. So we're just going to go through numbers 1 through 7. And it begins with a synapse, a chemical synapse at the motor end plate. So we'll see on the right, we have an action potential. So this is an axon of a motor neuron that behaves just like the axons that we've detailed earlier. So just imagine calcium flooding into this axon terminal, that causing the release of a neurotransmitter that are packaged in synaptic vesicles. We have a dumping of that neurotransmitter. In this case, that neurotransmitter is acetylcholine. What happens then? Step 2, acetylcholine triggers what are called nicotinic receptors. All right, so let's focus in on one of these little finger-like projections at the motor end plate. That's what we have here. And the cell membrane in the motor end plate is packed with these nicotinic receptors. And they're similar to other ligand-gated channels that we've seen before, with some distinctions. So first of all, they bind to acetylcholine molecules. That causes them to open their gate, which allows for ion flux. And the ion flux that is permitted is a little bit different than some of the others that we've considered. First of all, potassium is allowed to leak out of the muscle fiber. So what is that going to tend to do? It's going to tend to hyperpolarize the membrane, which you would think would be the exact opposite of what we're after here, because we want to excite the cell. But at the same time that a small amount of potassium is permitted to escape, even more sodium rushes into the cell. So the net effect is to depolarize the cell, and that creates a depolarization that then propagates along the surface of the cell. So if we look above in this schematic, step 3 is that the action potential propagates along the membrane. So if we look up here, we can see an animation of that excitation. Okay. So what does that do? Well, that brings us to the next step, which is what happens in the interior of the cell. Along the T-tubules, there are these proteins called the dihydropyridine receptors, or we'll just call them the DHP receptors. The DHP receptors have a protein that extends from the DHP and is mechanically coupled to this guy, the ryanodine receptor. The ryanodine receptor is a gate for the contents of the sarcoplasmic reticulum. So this helps explain why there's that close spatial association between the T-tubules and the SR, the sarcoplasmic reticulum. And when the action potential arrives at the DHP receptor, it activates the DHP receptor to pull on this stiff protein, which yanks open the gate on the ryanodine receptor. All right, so what we have in step 4 here is DHP plus RYR releases, in this case, calcium from the SR. Okay, so we'll animate that. So we have an opening of the ryanodine receptor. Calcium at this moment is at low concentration in the interior of the muscle fiber. So calcium will move into that space. It'll leave the SR and therefore elevate intracellular calcium concentrations. All right, and just like everything, well, almost everything we talk about at a molecular scale, you can imagine all of these components happening many, many, many times. There's many sarcoplasmic reticula, many T-tubules, and lots of DHP receptors and ryanodine receptors. So when an action potential arrives, just imagine thousands upon thousands of these DHP receptors getting activated, resulting in a large-scale release of calcium into the interior of the cell. Now we're going to talk about how the calcium activates the thick and thin filaments to generate contraction. And in that respect, it's kind of reminiscent of a presynaptic axon, in that the calcium in that case was activating motor proteins to drive the release of neurotransmitter that were packaged in vesicles. Here we have motor proteins that are activated by calcium again in the interior of a skeletal muscle cell. We'll get into those details in a moment. But what I want you to think about now is the fact that in order to control force, not only do you need to activate the machinery for generating tension, but you need to deactivate it. There has to be some means of modulating it so you can generate light amounts of tension or really strong contractions. And that amount of control requires not only that you flood the cell with calcium to create high tension, but also that you can remove it. And so another feature of the SR is a calcium pump that works in opposition to the ryanodine receptor. Okay, so that's what we see here. This is the sarcoplasmic reticulum calcium pump. It's active all the time. It snatches up calcium, any calcium that's in the interior of the cell. It's going to move it against its concentration gradient, which of course is going to require energy. So that's where we have ATP that just circulates around the interior of the cell, providing a source of chemical energy. It also happens to pump a proton in the opposite direction, which helps offset that +2 valence of the calcium ion. And boom, the pump does its work. It hydrolyzes ATP, that converts it into ADP, and it resequesters our calcium ion. And of course, this happens many, many times over. I think we've got thousands of calcium ions that are getting pumped back into the SR almost as soon as it's released by the ryanodine receptor. Okay, so that's step 4. All right, what happens next? Now let's focus on how calcium activates the thick and thin filaments. First, the thin filament, which we see in this reddish color. The thin filament is composed of G-actin. It's a string of G-actin monomers. So each of those circles there is a single protein, G-actin. It has a binding site for another protein we'll get to in a minute. This is almost like a beaded necklace, but the thin filament is composed of 2 beaded necklaces that are wrapped around each other to form a helical shape. So we're looking at just a half twist of 2 of those G-actin polymers. In addition, the thin filaments includes the protein structures that serve to control the tension that's generated by myosin. Okay, this is where the calcium comes in. The calcium activates a protein called troponin. Troponin is mechanically coupled not just to the G-actin monomers, but also to another protein known as tropomyosin. Tropomyosin sits on top of the binding sites of the G-actin monomers, and thereby blocking their access from any protein that might want to associate with them. Okay, so we've got a binding site, but that binding site at rest is blocked by tropomyosin. Now, the protein that wants to bind to the G-actin monomers is known as myosin, and the thick filaments, which are shown in blue down below, are composed of many, many myosin molecules, all of which have this almost like golf club shape and an elastic neck. Okay, so you've got this sort of bulbous head to the myosin and a springy neck, which you see up above. And the myosin is the motor that generates the tension. Okay, the thin filament, its most interesting feature is its ability to regulate the access of the myosin heads to those binding sites. So troponin, tropomyosin. But then myosin does all the work. As soon as it has access to the thin filament binding sites, then it will start to generate tension. All right, so let's talk a little bit more about how troponin, tropomyosin regulates this access of the myosin head. So let's just imagine the troponin molecule. Right now, the myosin is blocking the binding sites. Let's say the troponin binds to a calcium ion. In response to that, it will change shape. And it essentially, when it changes shape, it pulls the myosin out of the way, which makes the binding site accessible. If you look at that in cross-section, so we're taking a slice through two G-actin monomers with their binding sites blocked by a cross-section of two tropomyosin molecules, we can see that the troponin serves to essentially rotate the tropomyosin out of the way once it's bound that calcium ion. All right, so what's next? Now the binding sites on the thin filaments have been exposed. And what happens with the myosin molecule is it will bind to that binding site and begin to generate tension. And it generates tension in this cyclical fashion. So we call this the myosin cycle. That's the last step here. But the myosin cycle has lots of steps of its own that we're going to go through. All are in the service of binding to the G-actin and ratcheting along, sort of climbing along the length of the thin filaments. Okay, so we're just going through a cycle. We're going to start at 12 o'clock and we'll go through in a clockwise direction. So initially, we've got the myosin head. Now, each myosin molecule actually has two heads that are both capable of doing the same thing. We're just going to focus on one of them here. And initially, we're at this state where it has bound both adenosine diphosphate and inorganic phosphate. So ADP plus P. All right, so that's, of course, the product of hydrolyzing ATP. Okay, so that will be what happens at the end of our cycle. We're sort of focusing on what happens right after that's occurred. And that springy neck to the myosin molecule serves to store elastic energy. It actually functions like a spring that you have pulled. It's just that it's a rotation spring. So just imagine bending a spring, and if you let it go, boing, it will release stored elastic energy as kinetic energy. And that's what happens for the myosin head. So at the beginning of the cycle, as we go from 12 o'clock to 3 o'clock, we have the release of inorganic phosphate and the start of what we call the power stroke. This is when the thin filament is moved relative to the thick filament. And so it looks like that, and you can see the motion there. So the myosin, that springy neck, is causing the myosin head to rotate and sliding the thin filament leftward. Now, that's the beginning of the power stroke. At the end of the power stroke, we have the release of the ADP molecule to complete the power stroke. So what's left as we go from 6 o'clock to 12 o'clock is all of what's needed to reset the myosin molecule. So that includes binding an ATP molecule. When ATP becomes available, then the myosin molecule will detach from the thin filament. The myosin will then hydrolyze the ATP to produce the ADP plus the inorganic phosphate. And what you could see there up at the 12 o'clock, you can scroll back if you want to repeat it, was a reset of the myosin molecule. So if you compare 12 o'clock position to 9 o'clock position, you can see that there's a subtle difference in the shape of the myosin molecule. That's because when the spring is essentially loaded, then the myosin molecule rotates forwards to a new binding site on the thin filament. Whereas at the end of the power stroke, it's in that sort of leftwards configuration. All right, so there are a couple things I want to point out here about the myosin cycle. First of all, because of that elastic neck in the myosin molecule, we have a difference in time between when the force is generated. The force is generated between 12 o'clock and 6 o'clock on our diagram here, but the energy is actually cashed in between 9 o'clock and 12 o'clock. That's where the hydrolysis of the ATP occurs. So there's a decoupling in time between use of or exhaustion of chemical energy and the creation of mechanical energy. Another thing that's interesting is rigor mortis. So when somebody dies, their body very quickly becomes quite rigid. And that can be explained with respect to the myosin cycle, because that rigidity occurs because the myosin heads in all the skeletal muscles of the body fail to dissociate with those binding sites on the thin filaments. And the reason why is because when you die, your body fails to circulate substrates for the production of ATP, such as glucose or fatty acids. And so you have a big drop off in ATP within a cell. And if ATP is not available, then you have no dissociation of the myosin heads from those binding sites. And as a consequence, muscle can be quite stiff if you don't have this ability to detach myosin from actin. Okay, so we've stepped through the myosin cycle and all 7 steps before that. In summary, we had the synapse at the motor end plate, the acetylcholine binding to the nicotinic receptors, thereby exciting or creating depolarization in the muscle fiber. That action potential propagates along the membrane for the cell. That in turn activates the DHP and ryanodine or RYR receptors, which release calcium from the SR into the interior of the cell. That calcium in turn activates troponin, which moves tropomyosin out of the way. That exposes the G-actin binding sites, which allows for the myosin cycle to occur. We call this whole process Excitation-Contraction Coupling, or EC coupling. And in a future lecture, I'll contrast all of these events for EC coupling, which apply only to skeletal muscle, although there are some similarities in both cardiac and smooth muscle. But everything I've told you so far is only for skeletal muscle. In a future lecture, we'll talk about how it's different in those other 2 categories of muscle.